You're unmuted.
Hello, and welcome to the 2025 Environmental Science Honors presentation of their field research experience at the Headwaters, with Headwaters. The students went to Rodeo Lagoon in the Marin Headlands, and I'd like to introduce M.K. Schumacher from Headwaters Institute to talk a little bit about the program and the presentations that the students are going to give. So thank you.
Hello, everyone. Nice to see you via stream. I had the pleasure of working with all of these students over the past 2 weeks. We started in class with a question asking and methods development class. And then, like Mr. Vann said, we went out to Rodeo Lagoon and collected a lot of really, really good data. And then they came back into the classroom, analyzed that data, and started their presentations. So I am very, very excited personally to watch them present on their findings. They put a lot of hard work into these presentations, so we're very excited to watch them. So without further ado, we'll have our first group come up.
Do you guys want to I'm not.
Okay, should we stand?
So you can see yourself right here.
Okay.
It's okay if not all of you are in it, but you'll just progress the slides by doing this. Okay.
Okay, cool.
Good luck.
I'm Dylan.
I'm Oscar.
I'm Evan.
And I'm Alex.
And we did the effect of distance from lagoon on soil moisture level and plant VPD.
Yeah, so basically on the slides, we'll be going over kind of the introduction to our kind of study, and then the materials and methods we used, what we kind of found after gathering the data, and kind of discussing the data and any potential errors and caveats we have. So this is the introduction, and our research question was, how does distance from the lagoon affect soil moisture level and the VPD of plants? To touch more on both soil moisture and VPD, soil moisture, as you might be able to infer, is the water available in the soil. And it's really important for both water absorption and nutrients. And oftentimes low soil moisture leads to struggling to get water and nutrients, and also means that you're more vulnerable to different natural events. And then VPD is something a little bit more unique. It measures the vapor pressure deficit of a plant. And basically that tells us the difference between how much moisture is in the air and how much a plant can actually hold. And this is kind of important because it tells us how much water is being absorbed by the plant and its transpiration. So high VPD means that the air is very dry, and it means water is leaving really quickly. And low VPD means there's humid air, and it decreases the amount of water vapor from leaving the plant.
Okay, so we had three variables. The independent variable for both of the dependent ones was distance from the lagoon. We measured it in feet on the site because we did not have access to a measurement in meters, and then converted it later. The first dependent variable was moisture content. Is measured on a scale from 1 to 10 using a 3 tool. And then the dependent variable, the other one was the VPD level, which is measuring KPA, which Oscar just talked about. So our hypotheses, first, the hypothesis was that as we get closer to the lagoon, we would expect that the soil moisture would increase because the water from the lagoon would be absorbed by the soil, and there'd be more of it since it's closer to the lagoon. And then the VPD level would decrease because surrounding area is likely more humid, which would reduce the vapor pressure difference between the leaf interior and outside. And then the null hypothesis was just that there would be no significant decrease, no statistically significant decrease or increase from the for VPD and soil moisture levels based on proximity to the lagoon.
Okay, materials and methods.
First, we'll kind of introduce, this is how we measured VPD, and it's using a vapor pressure deficit gun, which basically just measures the amount of moisture or vapor leaving the plant.
Okay, we also use the soil moisture meter, and this gives you the amount of water in the soil, as Oscar said, and you kind of stick it halfway down into those prongs into the ground, and it gives you a reading, 0 being really dry and 10 being really wet. And then this is a transect. It's basically like a massive measuring tape. Ours went up to 100 feet, and we used it to measure distance from lagoon. Okay, methods. We measured out 30.5 meters, which is 100 feet from the edge of the lagoon with the transect. So basically Alex would hold it at the edge of the lagoon and we'd walk backwards. And starting at 30.5 meters, every 3.05 meters, we would select a random plant where we were standing and we would identify it. And then we'd stick the soil moisture meter into the ground to get the soil moisture reading, and we'd do that right next to the plant to see if there was any correlation. We would use the VPD gun to then take the VPD of our selected plant that we identified, and we would move forward another 3.05 meters and repeat this process until we reach the edge of the lagoon. And for our primary sample sites, we walked all the way up here for another group's data actually, and then we stopped by this marsh here and we got some higher soil moisture levels. Then we ate lunch and then we walked down here to get our 190 feet, 80 feet kind of data, and then we did the same thing there and there too.
Okay, so now we're going to talk about the results. So upon collecting the data, we inputted it into Google spreadsheets. These are all the variables up here. The distance meters and the moisture level and the VPD level are the three variables we focused on with our graphs. So each of the columns represents the variable and the rows represent each round of data collection that we did. Okay, so this is our first graph. This graph has three variables. This is kind of like the culminary graph. It kind of represents all the data we collected. So on the x-axis, we have the distance meters. So on the left side, that's closer to the lagoon. On the right side, that's way farther away. On the y-axis, we have VPD level in kilopascals. And so, yeah, higher up means higher VPD. And then our z-axis was, or z variable was moisture level, and that's represented through like a color gradient of each of the dots. And so with this graph, it kind of seems like based on a visual test that there's a loose, like loosely, as you get closer to the lagoon, VPD level and moisture level increases. But because it's a three variable graph, we couldn't do any statistical tests, which is why we did a few other graphs to show more precisely the correlations. So here is the effect of distance on the moisture level of the soil. Despite some mixed results, especially with inconsistent moisture levels of the soil within like 50 meters of the lagoon, generally it seems that the soil closer to the lagoon is more wet than farther away, which kind of makes sense. And then using a linear regression test, which was the statistical test which best worked with two numerical values here, we calculated the p-value to be less than 0.01, which means basically that there's an extremely strong, extremely significant, strong confidence that there is a relationship between the two variables. So it's not just due to chance, our findings. And then we used a cubic graph because that kind of best represented our data. But that cubic graph had an R-squared value of 0.467. R-squared value basically represents like how closely the model fits to the data points. So a low R-squared value like that indicates that there's not an exact pattern we can identify. So this is a similar graph, but this time it's the effective distance on VPD level. And this graph kind of had like somewhat similar results, but it had even a bigger variance. So it makes it really difficult to find the relationship between the distance and their VPD level. And we used a cubic graph again to represent the data because that's what kind of best fit our results. And using a linear regression test, we calculated p-value to be 0.13, which is significantly above that 0.05 line where you want to be to establish like a significant effect. So there's only like some indication that there might be a relationship, and we'll talk more about what that means. And the cubic graph had an R-squared value of 0.0525, which is really low. And so we can't really identify a pattern here. And then this is the effect of moisture level on VPD level. I labeled this as bonus because it's not actually the effect of independent variable on a dependent variable, but instead we did the dependent variables just because we thought it would be interesting to see if there's any relation here. We won't really talk about this, but yeah, so we kind of did the same thing here. We plotted it. We used a linear regression test, a cubic function best modeled the graph again, and we calculated the p-value to be actually 0.02 here, meaning there is strong confidence that there is a relationship between the variables. It's not just due to chance. And then we had another really low R-squared value, which means that there's not an exact pattern we can identify. But also with this kind of graph, we recognize that like this causation may be not actually like moisture level causing VPD, but it may just be the distance affecting both of them in the same way. So it looks like there's a correlation.
Okay, so now the discussion section. The first thing was analyzing the first graph, which was the effect of distance on moisture level. To summarize or recap, like our original hypothesis was that the closer to lagoon we'd be, the higher the soil level would be. So as Evan said, the p-value was less than 0.01, which means that there definitely is some statistically significant relationship between the independent and dependent variables. The p-value is essentially just like a metric of whether or not the differences are based on random chance or based on some relationship between the two variables. However, the r-squared value was 0.46, meaning that only 46% of the results can be explained by the independent variable, which is like lower than we want. Ideally, it would be between 0.5 and 0.7. So essentially what r-squared is this like a test of whether or not, or how much like the dependent variable can be seen as a result of the independent variable based off how closely they align with the line of best fit. So thus we can reject the null hypothesis because there is a connection between the independent and dependent variables as proven by the p-value. However, we cannot accept the null hypothesis because we cannot be sure that it's the independent variable which is causing this difference. And like Evan said, we use a cubic model, which means like we modeled the line of best fit as a cubic graph. So for VPD levels, doing a similar analysis, our original hypothesis was that the closer to lagoon we be, the lower the VPD levels would be. The p-value was 0.13, which is much higher than 0.01 as the other graph was, and it's above the 0.05 threshold for a significant difference, which means that we cannot be sure that the differences are based off like the independent variable changing, and cannot be sure that like it's not based on random chance. The r-squared value is also too low to establish a relationship between the independent and dependent variables, so we fail to reject the null hypothesis because we cannot be sure that like it's not based on chance. The reasoning we kind of explained for the first one, there was a relationship between being close to the water and soil moisture levels. However, like our tools are pretty inaccurate and use like whole numbers with like very low nuance, so they may have contributed to like the broad r-squared values. And for the second one, the VPD gun, which we'll talk about in the caveat section, was not very consistent, which may have contributed to that high p-value. To answer some of the other research questions that we had coming into this, current science is lacking in these fields in this region, so we cannot be sure like how our results compare to existing things, and it cannot really be used. Our results cannot really be used to guide future conservation efforts. And then Rodeo Lagoon has like unique circumstances because of like the brackish water and fog, which may have contributed to our data, as we'll talk about the caveats.
Yeah, now we'll kind of talk about the caveats and future research. So one of the biggest caveats we had was there's the possibility of inaccurate tools because the soil moisture meter, which was one of the main tools we relied on, had no units. So we weren't sure what we were actually measuring in. It just went 1 through 10, and we weren't sure how that actually measures how like moist the soil is. And the VPD gun, it also gave us varying results, partially because there was likely error on the human side, and partially because just the way it works, it might have picked up the vapor like not directly next to the plant, but maybe a little further away. Another caveat would have been distance imperfections. You always want to be kind of directly perpendicular to the lagoon, but because of like different plant growth, that was almost impossible. So we ultimately had to result in like kind of coming in diagonally to the lagoon. And then also, as we were measuring, plant species were inconsistent throughout the different locations we were trying to take data in. So that also would have contributed to potentially different VPDs because of like structural and biological differences that they have. And then finally, weather shifts would have also been a caveat because as the day goes on, it got warmer and less foggy. So that could have potentially affected both the soil moisture just from the sun drying out some of the surface soil, and then also the VPD. Okay, so some improvements we kind of made was that instead of choosing two dependent variables, we should have had one, because right now we have both the distance from the lagoon and soil moisture level and VPD. We also should have used more accurate methods just to estimate the soil moisture level, just because the soil moisture meter was very inaccurate. And then identifying one species of plant would have definitely helped in helping us keep the VPD and consistent.
And a future follow-up question we could have is, so because we learned that dry air leads to an increase in VPD and plant loses more water through transpiration, We know that the stomata closes, preventing further water loss, as we kind of talked about in our first unit. And then with humid air, it causes a decrease in VPD, and plant water loss through transpiration is reduced to make sure there's enough water in the plant. And the stomata opens, allowing water to escape to make sure there's not too much water. And then, so we know the opening and closing of the stomata is regulated by potassium ions. So we thought it would be interesting to explore how does presence of potassium affect VPD levels, because it controls stomata opening and closing.
Okay, we have like 1 minute for questions. If anybody has any questions? No? Okay. Then we have a quiz. I'll give you like 15 seconds per question. Name our independent and dependent variables in our experiment. Just like write it down. Hurry up. Yeah, okay. Shu, what was your answer?
I just got my pencil, but I said VPD is independent.
Okay. Anything else? Okay.
Any other?
Good answer. Okay, name all the pieces of equipment we use to collect the data. Shoot again.
Shoot.
Shoot, what was
Same
your?
thing, the circle, like,
Oh.
building the yellow button.
Okay.
Transect, yeah.
Yeah.
Do you know the other two?
What do we use to measure soil moisture?
Water vapor measure.
Last one, challenge question. If you saw, it was on one of the slides, name at least one plant species that we identified and collected data from. Nobody? Okay.
All right, no, that's fine.
It was the ice plant, which is also, the scientific name is Azoaceae. So yeah, cool. And then this is our bibliography. And then, yeah.
Don't forget to take notes on the presentation.
God damn it,
and we're ready for our next group. That was a group, let's see, I think Penny, John, Miley, and
How do I? Oh, we never.
you guys see this. You guys are welcome to begin.
Okay, this is brackish business, the effect of pH and moisture on species diversity by the four of us, and with Mary Ellen Benyer as our scientist. Okay, so our experimental question is, how do soil pH and soil moisture affect species diversity at Rodeo Lagoon, or biodiversity? So as you can see, our independent variables are soil pH and soil moisture. Our dependent variable is plant biodiversity, which we counted by just the number of unique species in the quadrat that we placed. And we're gonna get more of that into the methods. So first, some background information. Soil pH affects like H+ ion interactions with like different plants, which like then affects like which plants are actually able to live in certain areas. So plants will adapt to living in high pH environments or low pH environments, as Penny is gonna get into in the next slide. Which just means that different plants thrive at different pH levels, which is why we're trying to figure out like what the plants are most suited to at Rodeo Lagoon. And then the same thing is kind of true for soil moisture, where plants adapt differently to live in different soil moisture conditions. And so some will be able to live in more extremes, whereas some lower, like most will probably be able to live like closer to the middle.
Just to expand on that a little bit, you can see in our two figures how pH can affect how a plant is able to take up, or the nutrient composition of soil. So when there's, pH is basically a measure of the hydrogen ions in a substance. So at lower pH, there's going to be more hydrogen ions, and at a higher pH, there's going to be less hydrogen ions. So at lower pHs, macronutrients like potassium and sulfur are lost from the soil, and the plant can't uptake them. because the H+ ions bind to negatively charged soil particles and basically take up that space from those macronutrients. And the opposite is true of higher pHs, where micronutrients are not able to be used by the plant. So basically, plants will adapt to different environments based on the nutrients, adapt to different pH values in the soil. And the same is kind of true of moisture. You can see in our figure, plants that live in very moist environments and plants that live in very dry environments have a lot of different adaptations that make them more suited to their environment. So our goal with this study was to find out the ideal or the optimum range of pH and moisture for the plants at Rodeo Lagoon. Okay, the purpose of our study, like I said, is to identify soil conditions at Rodeo Lagoon that support the greatest plant biodiversity. And that's really important because biodiversity is really essential for ecosystems and their resilience to things like climate change, changes in their environment. If there's a really low diversity of species, then it'll be really hard for that environment to recover from things, from like events, from global warming, from changes to that ecosystem. So it's really important to understand and preserve biodiversity. Our null hypothesis for this experiment was that there would be no difference in species richness across pH or soil moistures. And our alt hypothesis was that the highest richness would be at intermediate pHs of about 6 and 7 and intermediate moisture levels.
Okay, so for our materials and methods, we use the 12-meter line transect, and then we use the 1 foot by 1 foot quadrat. And then we use the 3-way soil meter, which we use to measure the pH and the soil moisture. And it does have a scale from like 0 to 10. So we just did percentages. So instead of saying like 1 through 10, we would say percentages because we thought that would be an easy way to measure it. For our methods, we established, we established a database of all the species present in the area. So I was taking notes and we at the time we named them random names, but we then figured out their actual names. And then we laid the transect parallel to the shore of the lagoon. And we placed the quadrat every 2 meters. And then we recorded pH and soil moisture percentage with the 3-way meter. So whatever number showed up on the meter, we just converted that into a percentage. And then we identified and counted the different plant species within the quadrat and took notes on that. And we repeated that with 3 transects for 18 total samples. And we also went to 2 different parts of the lagoon. So we started off, we did 11 samples in one part of the lagoon, and then we did 7 in a different part.
Okay, so this is just some more on our materials and methods. So in figure 3, you can see that those are some of the plants that we identified, and we also have the real names and the field names. So like Miley said, we just gave them kind of random names at first just to be able to track them because a lot of them came up multiple times, and then we identified them later using some of the app that we were given. And then in figure 4, you can see us conducting the method. So the quadrat is 1 square foot, and we would basically just count how many number of individual species we saw within it, and then just like Miley said, record the soil moisture and pH. And then in figure 5, you can see just the different locations that we sampled at with the red dots.
Okay, now we'll get into our results a little bit. So as you can see, we chose to graph moisture and pH separately. We felt like a 3-way graph was a little bit too confusing, and we wanted to kind of understand their effects on species biodiversity differently. So the model that we chose to represent our data was a quadratic line, as you can see, which means that which is pretty aligned with our data set. Moisture and pH seem to peak a little bit more in the middle, though we do have pretty big error bars due to our limited sample size and our kind of inconsistent equipment. But the issue that we ran into with our data was that DataClassroom only does linear regression, which basically is analyzing how the independent variable impacts the dependent variable. whereas our data is more suited to a quadratic expression. So, but we had limited tools to work with, so we calculated our p-value based on a linear regression model, which put out some pretty high p-values of 0.97 and 0.68, which is a very high number, indicating that our data is basically just due to chance. But that also kind of reflects like a limitation of our equipment and our experiment, because we were unable to do other types of statistical tests that are out in the world just because of our limited knowledge, and DataClassroom doesn't have them. We did attempt to do some calculus and try to graph the derivative and other things, but it was unsuccessful due to our limited knowledge. So based on the tools in DataClassroom, yeah, our data is insignificant, but more qualitatively looking at the results, we can see there does seem to be a bit of a peak in species biodiversity at intermediate pH and moisture values, even if, according to our model, the data was not significant.
Right. So as Penny said, we cannot determine any evidence of significance because we have a p-value of 0.97, which means that we can't reject our null hypothesis. However, you could interpret it as the fact that it doesn't fit a linear line is actually proof that our hypothesis did work, because we didn't predict it would fit something linear. We predicted it would peak and then go back down. So I guess that's up to the viewer. But so we found no statistical significance. However, as Penny said, qualitatively, you can kind of see that there is a peak in the middle and that you see downturns on both the beginning and the end. We also noticed a peak near a pH of around 7, which is pretty high overall and doesn't seem to be like a super, like it seems to be higher than average in most ecosystems. However, as the figure shows on the right, basically, so the top figure is just the like sodium like increases in different ecosystems, and then the bottom one is pH increases in those same ecosystems, and it basically just shows that having more salt in the water increases the average pH of systems. And so because Rodeo Lagoon is brackish water and not freshwater, it makes sense that it would have higher than average pHs compared to freshwater ecosystems, which is why we got a pH of 7 as the peak, which is just a pretty high value in general. And then for soil moisture, again, we can't determine any evidence of significance because we have a P of 0.68, so we cannot reject our null hypothesis. However, again, qualitatively, I think biodiversity has an even stronger correlation than pH in our graphs, because you do see a pretty significant increase in the middle, but also we did have very limited sample sizes, and we had different sample sizes depending on the moisture content, because we couldn't standardize it. So we had very high error bars that still kind of did overlap at the end of the day.
Okay, so our findings suggest that Rodeo Lagoon species thrive the most in like moderately alkaline or moderately moist and slightly alkaline conditions. So this makes sense because the lagoon is influenced by brackish water, which can raise the pH. And it connects to our broader ecological theory because biodiversity is often highest when conditions are intermediate. And like conditions that we tested in specifically like soil moisture and pH, like the species will thrive best when those are at intermediate levels. And you can kind of see this illustrated in the figure that when too little or too much moisture decreases productivity in the species growth. And from a land management perspective, this means that keeping soil conditions in like a middle range, not too dry and not too saturated, could help preserve biodiversity in wetlands like this. And also there can be risks to this. So climate change can kind of tip the scales. And if temperatures raise and the sea level rises, it can lead to more like saline conditions, which would in turn make the conditions more acidic since the salt raises the pH of the water. And wetter soils could also stress the species that are not adapted to extremes. And in the long term, if pH and soil moisture shifts too far in either direction, we expect the biodiversity to drop. And only stress tolerant species would be able to survive. So there is also important limitations in this study. And as we stated before, we only sampled 18 quadrats across 3 transects, which is a relatively small data set and may not capture like full variability across the lagoon. And
the lagoon is pretty big and there is a lot of plant life around it. So also the equipment we use, a handheld 3-way meter is likely imprecise, and we noticed that the pH values varied quite a bit, as my partners talked about earlier. And this can reflect an instrument error, which it can reflect instrument error, but also reflect natural variability just as much. So the third is that our sample sizes weren't balanced across different soil moisture and pH categories, which kind of affects the accuracy of our statistical measures and the standard error of the mean. And then going forward, we can improve this by increasing the number of transects and to get a broader coverage of our area, and also sampling at multiple times of the year to account for seasonal changes, which definitely has an effect in a coastal environment like this. And just using more accurate soil probes to get more reliable data. And then finally, these improvements could kind of just give us a cleaner picture of our findings and our data and how soil really affects the biodiversity in the area.
Okay, so for future research, to follow up, we want to ask the question, how does soil salinity interact with soil moisture to influence plant biodiversity at Rodeo Lagoon? We think that the coastal wetland is impacted by ocean tides and salt spray, and then salinity alongside our independent variables determine which plant species thrive because some are more tolerant to it. And then why? Because we get a more complete picture of the environmental controls on biodiversity in ecosystems like Rodeo Lagoon. So this question is directly related to our study because Rodeo Lagoon is a coastal wetland, which is influenced by ocean tides and salt spray, and salinity likely plays a key role, how Riley said, alongside pH and soil moisture in determining which plant species thrive. So if we investigated soil salinity, it could provide a more complete picture of the environmental controls on biodiversity at the lagoon. Here's our bibliography. Does anybody have any questions? Yes.
How did you guys determine where on the lagoon to collect your data? Thinking about salinity and stuff like that as well.
It was mostly just based on accessibility for us, where we
There was
could
a lot
go.
of
There was a
poison
lot of
oak.
poison oak, so we were just kind of navigating where. But we did try to spread it out, like you saw on the red dots, that we had them kind of evenly spaced across.
Yeah, we also tried to get areas that would have a range of okay, there's kind of marshy plants here, and then there was sections of the environment, if that makes sense.
Yeah, it took us 30 minutes to find it.
So do you think if you could have gone to different places, more different places, across the lagoon, that would have changed your data at all?
I think having a higher sample size definitely would have improved our data, but I don't know beyond that how different would have been.
Okay. Anybody else?
I think you mentioned that to determine plant biodiversity, you measured the unique plant species. Can you talk about how you calculated that and why you didn't just include all plant diversity through the diversity index equation you can use?
Well, we had a quadrat, so we kind of just counted which ones looked different.
We identified the different species of plants, and then we counted how many.
I mean, the numbers were low. It was
species were
3,
in the
4, and 5. So I don't know how much an equation would have been necessary. We were able to species look different.
Okay. I don't think we have time for the quiz.
Yeah. Okay. Great.
It was quick to have it. Thank you.
Yeah.
Thank you, guys. We have up next Ella, Ryan, and Anya.
Okay, so this was our presentation. We titled it Water Under the Bridge because we are studying the effect of relative distance from the ocean on water quality, which is measured distance from the bridge. I'm Anya.
I'm Ella.
I'm Ryan.
I'm Alva.
And our advisor was MK. Yay. Okay, so our research question was, what is the effective location on water quality, which we measured in distance from the bridge, which we will give a map as to where this bridge is in relationship to the lagoon later on. Our dependent variable was water quality, which we tested using turbidity, dissolved oxygen, and bug life, as well as we were curious about various other forms of life around the lagoon, which we kind of more informally counted. Our hypothesis was that the water quality in the lagoon further away from the bridge will be higher. And our null hypothesis was that there would essentially be no correlation. Distance from the bridge will not affect the water quality. So a little bit of background. This is because the bridge is close to the ocean, water further away from the bridge would have less salinity. And increased salinity levels mean that there's a lower dissolved oxygen content because there's less room for oxygen in the water if there's more salt. Higher turbidity, which is if there's more dissolved particles in the water, it makes it more turbid, less clear, and overall less healthy water. And this has many practical implications for the lifestyle and living conditions of wildlife in and around the lagoon. So yeah.
All right, so here you can see where we chose to sample our data. We started, like Anya was mentioning, at this bridge. We wanted to kind of start closer to the ocean where the salinity would be slightly higher, but we also wanted to make sure that we were sampling in a location where there would be consistent water all year round. We didn't want the tides or seasonal variants to be an effect. And looking at this water over here, it seemed like that water could perhaps be more temporary, coming in and out. So that's why we chose to start at the bridge rather than closer to the water. And then from there, we measured 85 meters subsequently across the lagoon, and we were able to get in 5 different sampling locations where we did all of our identical tests, measuring all the 3 independent variables that Anya was talking about, independent variables. Okay, so here are our materials and methods. So we started by measuring the turbidity just because the other tests had the potential to kind of mess up the water a little bit. So we wanted to start and make sure that we were getting an accurate reading for turbidity. So for turbidity, we just took the big tube and we submerged it in the water right off the shore. And then from there, you just let water out of the bottom until you can see the design at the bottom of the tube. And then this is measured in centimeters. So the fewer centimeters means that the water is less clear, right? Because you have to let more of it out to be able to see all the way through it. So, when you have 10 centimeters, that's when there is higher turbidity. And then we moved on to looking at the macroinvertebrates and identifying the different bugs that we found in the water. So to do this, we took a D-net and we waded into the water, kind of stirred up the sediment and the soil a little bit, and then collected 3 scoops at each location and then put it in a clear plastic container. And then we kind of looked through this and identified different species of macroinvertebrates that we found. So kind of using magnifying glasses and then identifying them and matching them to a sheet that we had, which kind of basically correlated the different species of macroinvertebrates to different water qualities. So going from group 1 to group 4, with group 1 being species that can only survive in really clear and clean water, with 4 being species that survive in higher pollution. So by identifying the different species of bugs that we found in the water, we were able to correlate them to different water qualities. And then each of these groups are given a specific score, which allows you to calculate a total water quality score based on separate species of macroinvertebrates that we were able to find. So we gave each location an overall water quality score with the macroinvertebrates. And then finally, we measured dissolved oxygen. We took 3 trials at each location where we just filled a plastic cup with water from the lagoon and used the dissolved oxygen measuring device to measure the dissolved oxygen. And we took the average of the 3 trials. Yeah. All right, our results.
Okay, yeah. So these are our results. These are the first 2 graphs. The first one is the distance from the bridge and how that affects dissolved oxygen. And then the one lower than it is the distance from the bridge and how that affects turbidity. So for both of these graphs, they had pretty significant results. You can see that as distance from the bridge increases, the dissolved oxygen also increases. And as distance from the bridge decreases, the turbidity also decreases. And for the first graph, our R-squared value is 0.97, which is saying that there's a very good chance that the independent variable impacted the dependent variable. And then it's the same, it's 0.958 for the other one. Sorry. And And then for both of them, our p-value, well, for this one, for the dissolved oxygen graph, our p-value was less than 0.01, which means there's a really good chance that our data was not due to chance. And then the p-value for the other one was less than 0.05. So for both of those, we have pretty good data. And then these are our other two graphs. This is distance from the bridge and how that impacted the presence of fish and other wildlife. And then the other one is distance from the bridge and how that impacted the macroinvertebrates. So for fish and other wildlife, our data is a little bit more scattered, but it does indicate like an upward trend, which makes sense because as you go farther away from the ocean, there's more like ducks and other waterfowl. Yeah. And for the macroinvertebrates, we did not have too good of a p-value. Our p-value was 0.26. And it is supposed to be like less than, or if it's less than 0.5, then that indicates that there's like a strong correlation between the variables and that they're not due to chance. And ours was 0.26. So there is little to no indication of a relationship between the two variables. Yes, and then we also observed a sulfury smell at the 250 meter point, which is right here, which is kind of our outlier, if you can see, because we didn't find any macroinvertebrates. So that like could have been a reason, it could have been related, but we don't really know. Yeah.
Okay, so in terms of what all this data means, overall our results do support our hypothesis that the lagoon water quality further away from the bridge is higher. But where are dependent variables? Turbidity, dissolved oxygen, and macroinvertebrates. So let's go into what each of those does for this hypothesis. First of all, as Abba was talking about, turbidity decreases as the distance from the bridge increases. And what that means is because turbidity is the cloudiness and opacity of the water, there is an excess in sediment movement from the waves crashing into the lagoon from the saltwater overflow. And that just creates more sediment and dust that spreads out across the water. And when we tested that, the water that was closer to that site where the waves crashed in is where there would be more sediment and, I guess, lack of clarity in the water. As for dissolved oxygen, it increased as the distance from the bridge increased. And that is because of increasing salt concentrations closer to the bridge. And that is because of an effect called the salt out effect, where more or higher concentrations in salt lead to more spheres of hydration, which take up available water molecules that will no longer be able to dissolve more oxygen. And as for macroinvertebrates, as Abba was saying, because it was too varied and due to chance, our data did not support our hypothesis. And it was really fun though. As you saw in that earlier slide, the one called Our Results, there was a worm there. That worm like sucked its head out of its neck. I don't know how to describe it, but it was really cool.
Like a turtle, like, you know. Like,
yeah.
Yeah.
Like that. So our data is also scientifically backed according to a 2022 study by the EPA, where they investigated a bunch of different freshwater or bodies of freshwater across the country. They found that an increase in salt contamination in freshwater does lead to an overall decrease in water quality. And to be more specific to our dependent variables, Atlas Scientific showed that an increase in salt concentration led to a decrease in dissolved oxygen specifically. And the National Park Service showed that the salinity and turbulence from the ocean negatively affected freshwater ecosystems. Overall, what this means for land managers is that there needs to be an effective, there needs to show that separation between the ocean and the lagoon is necessary to preserve the water quality and the species of those areas. Okay, and finally, to go into the nuances of Rodeo Lagoon specifically, as you all know, Rodeo Lagoon is very close to the ocean, and that is why there's a huge amount of saltwater contamination in the water. Without those factors, we would not have seen the same data, as there wouldn't have been those increase in turbulence and salt concentration. Additionally, Rodeo Lagoon is used in a lot of different ways by humans, from fishing to boating to just whatever, vacations. And that can also influence the way that we see the data in terms of macroinvertebrate presence and turbulence and stuff. And what this means is that as climate change worsens, the sea levels rise, and that will lead to an increase in saltwater and the waves affecting the lagoon, overall decreasing the water quality through turbulence and salt concentration and surviving bug species.
Okay, so here are a couple of our caveats that we kind of observed from doing our tests. First, the order of the tests. So basically, as I mentioned, we wanted to keep the turbidity happening before the collection of the macroinvertebrates, and then this was slightly inconsistent, as it mentioned in the second sampling location. So just make sure that we keep the order consistent. That could affect turbidity readings and also macroinvertebrate findings. And then also we could have made errors in identifying the macroinvertebrates. So we were looking at like different identifying factors like the amount of legs, the size, the different patterns, all that kind of stuff. But obviously they're really small and it's hard to miss. I mean, it's easy to miss really subtle differences and identify them with the sheet. So we definitely could have made errors there, which could have changed our overall water quality score at each of the locations. And then also we were obviously only able to test on one side of the lagoon. We were only able to test 5 different locations. And so that obviously is a pretty small sample size, and therefore by increasing that sample size, we can increase the validity of the data. And that could have been a caveat in terms of different variances that we saw.
Yeah, so kind of building off of that, for further research, we'd like to go like more holistically as like a follow up to like more to like exploring the relative distance from the ocean further into the lagoon, because like Ella mentioned, we were only able to get like a small sample set. Ideally, we would have gone like all the way to the other side of the lagoon. We also wanted to explore the long-term effects of overall discrepancies in water quality across the lagoon. So let's say that there is like lower water quality near the ocean and then better water quality like further away from the ocean. We wanted to explore like how that would affect different variables such as like macroinvertebrates and other wildlife. And so, yeah, we just wanted to dive a little bit further into some of those research questions. Here's our bibliography. Any questions? Great.
I don't know if you mentioned it, but like, how can you quantify like dissolved oxygen is like good or bad? Is there like a range DO should be in for species in water?
Yes, there is a range. I'm forgetting on what that range is, but we like Ella mentioned, we did use like the water quality, the dissolved oxygen meter that did give us that value. I forget what the exact range is, but yes,
When
there's.
making the graphs, we more just assume that the more dissolved oxygen obviously allows plants and all that like species that need oxygen. So we just kind of just assume that would be an indicator of like healthier water in terms of allowing plants and other species to grow. Obviously, like they can get to an extreme level, but we didn't observe too much like algae or like things like that. So we kept it pretty just like looking at it in terms of higher resource levels, like in the physical connection. Yeah.
I think you guys had a graph that was talking about like fish and birds and things like that. How
Yes, we
did you, like quantify that? Like,
So-
fish? How did you?
That was our kind of like bonus data because we had a bit extra time. So we looked at the area where we were in to collect the data, and like in our like vision, how many different like ducks / birds things that we could like see, we counted. And if it was more than 20, then we just put more than 20. Yeah. Yep.
Not like
Yeah.
one of our main like.
No,
I think we
this
saw
is
like
our
one
bonus.
fish.
Yeah, we caught one fish. Yay.
Quiz.
Any other? Wait. Okay. Let's quickly go to the quiz.
Yes.
So do you want to go through that?
Okay. You guys can just write this down and I'll give you like 10 seconds. What's the ideal R-squared value? Or the R-squared value that shows that your data is the most, like the independent variable affects the dependent variable.
And then a decrease in dissolved oxygen directly correlates to which of the following, based on our experiment. And then
So just write down the answers, 1, 2, 3.
what is in Rodeo Lagoon?
And then when you're ready, you can raise your hand.
If anybody wants to answer number 1.
You're gonna answer for number 1. Oscar.
1.
Nice job.
Yay.
Who has the answer for number 2? Sean.
15.
D.
No.
I don't think so. No, it's not D.
Closer to the bridge, not less dissolved oxygen.
Yes, but which one directly correlates? The distance from the bridge is a measurement of the salinity. So technically, it would be the salinity.
Yeah.
But if you're looking at the direct relationship, it would be the same.
Okay,
Riley?
and number 3.
Wait, Riley?
Increase in salinity.
Yeah.
Yes.
There you go. Number 3.
Number 3. No, it's
yeah.
not
No, that's.
Not you guys.
D. Okay.
Yeah.
Drink the water.
All right. Thank you.
Thank you.
I want to acknowledge too that Braden and Shu's partners are unable to be here today. So I want to thank Braden and Shu for carrying the weight of today's presentation. We'll hear from their partners later. All right.
All right, so this is our presentation on water quality and macroinvertebrate diversity. And so we are measuring what's the effect of distance from the most shallow, uncycled water on bug diversity. So our independent variable was the data collection site. So we basically just went to 5 different sites along the lagoon and took all our dependent variables, which was pH, dissolved oxygen, salinity, and then we also measured the macroinvertebrate diversity in their presence. And so macroinvertebrate population and diversity can be an effective indicator of healthy ecosystems, as you can see in the chart over there. So depending on which species are present, you can kind of estimate the amount of, like, how healthy the water is, basically. So our hypothesis is that as the distance from the shallow, uncycled standing water increases, the macroinvertebrate biodiversity value will increase due to better water quality. And our null hypothesis is that as the distance from shallow water increases, nothing will change. So essentially, we are first measuring the pH, which is showing how acidic or basic the water is. So too low of a pH will lead to difficulty of breathing, and too high will lead to ammonia becoming toxic, which reduces the available nutrients. And then salinity, it shows the dissolved salts in water. So too high or too low jeopardizes biodiversity as well. And dissolved oxygen. And so too low will kill sensitive macroinvertebrates, and too high will support biodiversity, but extreme levels can lead to oxygen crashes. So our methods was first to take a cup from the surface of the water and then measure, use our monitor and measure the pH and dissolved oxygen. And then, so our first measurement was directly next to the bridge at that first arrow there. And then we approximately went 85 meters along the edge of the lagoon and measured each site. So to collect the macroinvertebrates, it was a bit more difficult. We had to first use a net and basically do 3 movements back and forth with the net to agitate the dirt to get the bugs like in the water. And then we would scoop the water and dirt 3 times into a bucket and then separate it from there. And then we would put the different species of macroinvertebrate into the like an ice cube maker just to make it easier to see which ones are actually present. And then based on which ones are present in this chart, we could determine how good the water was. Yeah.
Okay, so essentially the results, we decided to display it through a bar graph. So 5 different sites, and we eventually found that there wasn't really a consistent upward trend in biodiversity based on the distance from the bridge. However, the ANOVA statistical analysis showed us that our p-value is less than 0.01, meaning that the data collected was still statistically different. So in the end, essentially, the null hypothesis is supported. Biodiversity did not consistently increase with distance. These are the 3 graphs. And so essentially there was no major difference between trials 1 and 5, and there was a lot of variability. So as you can see, there was a positive correlation between pH and bug presence at site 4. There was a big sulfur smell at this site, which made it kind of weird. So it kind of skewed up the data a little bit, but it was really cool to just see how the pH just kind of like, and the pH and bug presence kind of just like dropped based on that. So there are a couple of reasons for this. One is change or the lack of change. So water quality remaining similar with trial number, possibly due to like the season or dwelling location of the macroinvertebrates. More research required because obviously we did it in one day, didn't test different weathers. So yeah, also possibly due to the lack of biodiversity within the lagoon. Like obviously we kind of saw the same bugs everywhere. More research is probably required. And yeah, so essentially some findings were cyanobacteria visibly decreased as we went further and further away from the standing water, and other factors were more variable. Again, site 4 had the sulfur smell and the low pH, and we actually found out, or like sort of kind of made a guess on what the reason could be. So we think it's probably due to decomposition, so high levels of anaerobic decomposition from an excess of like waste, pollution, littering, low DO levels, etc., which leads to the production of organic acids and then thus will lower the pH. Yeah, so the soil was finer, leading us to hypothesize that these effects have been around for a while. Low pH, high decomposition, breaking down the soil. It's also possible that there is a hypoxic niche in the water underneath the surface, and only the surface of the water had high DO. So a lot of variability. We did not expect it when we made the hypothesis. Comparing it to similar data in pH, as you can see over here versus right here. So we can just see the times. Yeah, honestly looks pretty similar. Comparing the DO data, you can see a little increase right here. Here it sort of dips down at 8 a.m., goes up a bit. Yeah, so that's also another. Yeah, so this graph, honestly, is just the DO, or I guess like we would write down the DO after like 3 times of like measuring it, and then we would average it out and sort of put it for each trial. And as you can see, that is kind of the increase. So this is kind of like the main takeaway. What should we tell the people who are managing Rodeo Lagoon? We think that they should look at Site 4 for possible pollutants. There's a sulfur smell there, and there's not many bugs, pretty low pH. They should probably look into that and just kind of understand what's going on there. I think it could also tell us kind of like the future of the lagoon and the effect and impact that human interaction, i.e. like the people like surfing and hanging out at the beach, have on the area. So I think we should also take more macro invertebrate diversity readings. There was not much diversity in ours. So just a little background, there was this really tiny bug. I think it was called like a scud, right? It was everywhere. We only saw that bug. We saw a dragonfly larva, I think one time. We saw a few other bugs, but like it was mainly just this bug, showing that there was like a low amount of diversity when there should be other types of bugs like around the lagoon, but we didn't see any. Also indicating that these ones mainly are the ones that like survive given the conditions. So we also think that they should run more controlled tests to figure out how to increase the diversity of macroinvertebrates, and just like revitalize the ecosystem. So several factors, many sources have reported on large algal blooms occurring in the Rodeo Lagoon. That's not very specific, but it's still an impactful thing. The culprit, probably the people at the beach and their pollution. So the Marin County says that there's possible oil contamination. They didn't cite a reason. It is, however, pretty far from Sausalito, and the wind blows over the lagoon and from the ocean. So that could also be an effect because there's a lot of tourists there and a lot of beach activities. So here's kind of like the future. In 7 years, the DO has increased a significant amount, but the pH has stayed relatively the same. The DO increasing is unlike what should occur with rising global temperatures, but again, it was kind of a hot day, so it might just be that. As sea levels rise, the lagoon will flush easier with high tides, and salinity will increase, and eventually this water will sort of like start rising, get above the sand of the Rodeo Beach. And unfortunately, the lagoon will probably not be the lagoon we saw a week ago. The pH of the bay is much lower, so the lagoon's pH will also decrease. Yeah.
So there are several caveats, including limited data, changing time values, and data taken only on the coast. So yeah, as Shub said, we only took 5 trials, and they were along one end of the coast. And so if we went further along the coast or maybe to like deeper into the water, the data could definitely have changed and we could have come to some more concrete conclusions. But in the future, we'd collect more data points from around the entire lagoon. And yeah, so we would also test the impact of date and time on water quality. So we were doing these tests like one at a time, meaning like the time of day could have had a big effect on the water quality. And so if we could do it again, we would potentially take readings at the same time with multiple groups, or just test the difference of like one site and over time, like see how it changes.
Yeah, and like sort of adding on to that, if you go back just like a few slides to the sort of, or yeah, here I can, like over here, basically it'll show the time and how it's affected by these two. So it is a pretty big factor and something that like we didn't fully think about, but something we could really focus on next time we go.
Mm-hmm. And so here's our bibliography. And then, yeah, does anyone have any questions?
How do you think the oil contamination you talked about like affected your variables?
Great question. I think that the oil contamination probably pushed out a lot of macroinvertebrates. Also probably interacts with these nutrients, kind of contaminates it, causes like the good stuff to diffuse into the oil, so that the macroinvertebrates can't get it. Yeah, I think honestly, oil is a big problem in our environment. And
yeah, like adding on, it definitely affects the like at least the water quality, and it changes the ecosystem. So some of the existing species will like no longer be able to adapt.
Like the fact that there's like certain bugs that we should have seen aren't there probably shows that like many of these bugs cannot survive in oil. Only like scabs and a few others.
I would also like to point out the fact that those sheets are for freshwater organisms, and we were in brackish water. And so with that salinity, you're not going to see as many of those freshwater organisms as well. So it could be due to those pollution, but it also could just be due to the high salinity.
Yeah.
I just saw, well, you guys compared your graphs to graphs showing like the date and the time. I just wondered, do you think that was what affected your dependent variables more than the site location, which was your original independent variable?
Honestly, I actually think it did, because we weren't really able to measure like too much of the lagoon itself. Like we were only able to measure probably like 50 or to 25% of the actual lagoon. So I think that that could have had a bigger effect than the actual location.
Yeah, we probably like would have spent like in the future probably spend more time going around the entire lagoon. Like we really only went to like a fourth of it. So yeah.
You kind of mentioned that algal blooms could have affected like your results, and you said it was probably because of beachgoers. Can you kind of correlate how algal blooms be affected by beachgoers?
Sure. I think that beachgoers probably like will increase the killing of these plants and the killing of like certain nutrients and sort of like, yeah, things that are needed for these animals and macroinvertebrates, and that causes algal blooms due to like sort of the industrialization of the places around the lagoon and the agricultural runoff, I would say, contributes to that.
Yeah,
So,
and then
like,
the algal blooms will like affect the dissolved oxygen, and that would have a very negative impact on the lagoon itself.
Yeah.
Do you have any hypotheses as to why you think like the DO would vary so much throughout the day?
Yeah, I mean, I'd say like probably temperature and the tide, and like just sort of like, or yeah, just how the water is kind of affected by the time of the day and just like the heat. Like obviously we started off in the morning and it got a little bit hotter as we went, and that also changed the DO results. So yeah, I think water and oxygen levels increase from the heat. Yeah.
Your original hypothesis was like distance from shallow uncycled water, and then I think you sort of just measured it as like distance from the bridge. So do you just like assume to close to the bridge?
So we started off there and it was like really, really shallow just at the start of the bridge. So we kind of just, that was our checkpoint. Then we went 85 meters from there, 85 meters from there, and sort of had that as like our sort of main point for the shallow uncycled water.
Yeah, it was uncycled because compared to like the other water deeper into the lagoon, that's like it's deep and water's just being able to like diffuse and mix around. That water is like kind of more isolated compared to anything else. Yeah.
Okay, you ready for a quiz? Okay. What were our independent variables? Loudly.
The data collection site.
Okay, what could have been the cause of the sulfurous smell in trial 4?
Anya,
You said pollution potentially.
of course. And pollution by who?
Decomposition.
Who pollutes?
Oh, beach goers.
Yeah.
Also decomposition.
The culprit.
What would we probably change the next time we do this experiment?
Go all the way around the lake.
And what else will we do?
What would change about the time?
Oh, you do it like at the same time.
Riley.
Take more tests on like different macroinvertebrates.
All right, thank you.
Thank you.
Thank you all very much.